Hello everyone, welcome again to the series of solved previous year's ICRPG examination, Plant Science, where we will discuss the questions specifically on genetics and plant breeding. Today we will discuss the question paper of 2016, so let's get started. Question number 1. The purine and pyrimidine pairing in DNA is as follows. We know that the purines are adenine and guanine, and the pyrimidines are cytosine, thymine and uracil in case of RNA. So the correct answer is option number C, AT and GC. A, adenine and thymine, they are having 2 hydrogen bonds, whereas guanine and cytosine, they are having 3 hydrogen bonds. Question number 2. The cell cycle passes through the following stages. So the stages are gap 1, that is the growth phase, then the synthesis occurs, then the growth and preparation of the cell for mitosis, later the cell division. So the correct answer is option number A, G1, S, G2 and then the division phase. Question number 3. The biggest organelle of plant cell is, so it is the chloroplast, which is having the dimensions of about 5 to 10 micrometer long. Question number 4. RNA molecule which acts as an enzyme are called as, so they are called ribozymes. These ribozymes, they show a catalytic activity for the synthesis. Question number 5. Energy currency of a cell is, so it is adenosine triphosphate or ATP. ATP is formed by a nitrogen base called adenine having a ribose sugar with 3 phosphate groups. Question number 6. Chromosomal theory of inheritance was proposed by, so it was proposed by Sutton and Boveri in 1903. According to this theory, genes are the units of heredity and are found in the chromosomes. Question number 7. Who coined the term cell? So it was coined by Robert Hooke. While observing the cork cells, he found those compartments and named cell. Question number 8. The term heterosis was coined by, so it was coined by Schull in 1914. Heterosis is a phenomena where the hybrid progeny has superior performance as compared to their parent inbred lines. Question number 9. The number of characters selected by Mendel for his study was, so there were 7 characters, namely the stem height, flower color, flower position, pod shape, pod color, seed shape and seed color. From these characters, the contrasting traits were selected and then determined the inheritance. Question number 10. Synaptonemal complex appears during, so it appears during zygotine phase. It was discovered by Moses in 1956. Synaptonemal complex, it is a high-ordered protein associated structure which assembles between the aligned homozygous chromosomes in the mitotic prophase. Question number 11. The second generation Bt cotton contains genes of, so they contain genes that is Cry1AC and Cry2AB. These genes code for toxins against bollworm. Question number 12. The enzyme involved in transcription is, so it is RNA polymerase. It copies a DNA sequence into an RNA sequence. Question number 13. The total number of codons are, there are 64 number of total codons. We know that on a strand there are nitrogenous bases. There are 4 nitrogenous bases and 3 sets. So accordingly, there are 64 codons. Out of those, 61 they code for specific amino acids, whereas the 3 they are the stop codons. Question number 14. During DNA replication, the enzyme which separates the 2 sides of the helix is called, so it is called DNA helicase. DNA helicase, it breaks the hydrogen bonds between the complementary bases, which helps in unwinding. Question number 15. Barbara McClintock was awarded the Nobel Prize in 1984 for the discovery of. So she discovered the jumping genes, which are also called transposons. She saw them in maize. These genes, they are sequences of DNA that move or jump from one location to another in a genome. During cell cycle, intensive cellular synthesis, division of mitochondria and chloroplast, increase in energy stores, and formation of mitotic spindle will occur in. So it will occur in G2 phase, that is the gap 2 phase. Question number 17. The first meiosis in the embryo sac mother cell of plants results in. So it results in 4 haploid female gametophytes. We see the megaspore mother cell, the meiosis occurs, which gives rise to 4 haploid megaspores, out of which only one functional megaspore is present. Question number 18. The first miracle rice IR8 was produced in 1966 at IRRI Philippines through a cross between, so it was a cross between D. Jiu Wu Jin and Pita. This gene is from Taiwan, whereas Pita is from Indonesia. Question number 19. Crop varieties are classified as homozygous variety and heterozygous variety. One of the following is a heterozygous variety. So it is a composite variety which is produced by mixing of seeds of several outstanding lines which encourage open pollination to produce the crosses in all the directions. Question number 20. The hypothesis which explains heterosis as a result of complementation between divergent alleles is, so it is by the overdominance hypothesis. It was proposed by East and Shull in 1908. According to this, we have a combination of more divergent alleles which have better heterosis. If we see the combinations A1, A4, A1, A2, A2, A3 and A2, A4. So the A1 and A4 is highly divergent. It will have the highest heterosis, whereas A1, A2, A2, A3 and A2, A4, they will be equally vigorous. Question number 21. A form of apomixis in which the embryo sac develops from a vegetative cell of the ovule. So the correct answer is option number A, apogamy. Next question. The minimum progeny population size allowing for random union of all gametes arising from AA, BB, CC parent is. So we'll see that the progeny population size is given by 4 raised to power n, where n is the number of heterozygous population. Here we have 3 heterozygous population, so n is equals to 3. This implies the answer is 64. Next question. During substitution mutation, transition is caused by the factor. So it is caused by tautomerization. Tautomerization is a spontaneous transfer of 2 protons from one strand to another in AT and GC pairs that generates tautomers. Question number 24. During the process of transcription, an mRNA molecule is, so it is transcribed into DNA. Bulk breeding accomplish inbreeding in segregating generation. If there are 5 segregating genes in a population, near homozygosity to 100% will be achieved by, so it will be achieved by the F10 generation. Question number 26. The non-allelic interaction between or among genes controlling a trait is called. So it is called epistasis. Here the expression of one gene is modified by the expression of the other genes. Modified in the sense it can be masked, inhibited or suppressed. Question number 27. The hybrid progeny between a single cross and an inbred line is referred to as, so it is referred to as a three-way cross hybrid. We have a single cross which is crossed with the another line. F1 generated from this will be crossed by another inbred line. In case of double cross hybrid, we have two crosses. And the F1 generation produced from both the crosses will be intercrossed. In case of double top cross, we have a single cross which is crossed with an open pollinated variety. The F1 generation will be crossed with the open pollinated variety in case of double top cross. In case of multiple cross, we randomly pollinate in the selected lines. Question number 28. More durable insect resistance is found when the resistance is governed by, so the correct answer is option number C, cytoplasmic inheritance. It is the inheritance of organelle DNA in the offspring from the parent DNA. Question number 29. Among the different recurrent selection schemes, in all practical situations, the superior one was, so it was reciprocal recurrent selection, which was given by Comstock, Robinson and Harvey in 1949. Here, it improves two different populations in ability to combine with each other. The two parents, they act as a tester for each of them. Question number 30. A set of germplasm accessions derived from base collection to represent the genetic spectrum in the whole collection is called, so it is called core collection. Question number 31. The linkage arrangement in a dihybrid in which two dominant genes are on one chromosome and their corresponding recessive alleles are on the homologous chromosome. This type of arrangement of genes indicates a coupling phase where both the dominant alleles are present on one side and the recessive alleles are present on the other side. In case of repulsion phase, one dominant allele and one recessive allele is present, and on the other homologous chromosome, one recessive and the other dominant allele is present. Question number 32. Incorporation of two or more major genes in the host for specific resistance to pathogen is called, it is called gene pyramiding. Here, the stacking of multiple genes is there to a single genotype to combine desired traits through recombinant DNA technology. Question number 33. Under Plant Variety Rights Act, an extant variety can be legally registered and protected for a period of, so it can be protected for initially 9 years and a maximum of 15 years. Question number 34. Bajra Napier hybrids are an example of, they are an example of intraspecific hybrid between the Napier grass and the Bajra. Question number 35. In a living organism, a mechanism that can cause a gene to move from one linkage group to another is called, so it is called translocation. Question number 36. The first man-made crop triticale is derived from crossing, so it is derived from crossing of triticum and secale, which gives rise to a triploid triticale. Question number 37. A cell or organism having a genomic formula 2n-1 is called, so it is called a monozomic, whereas nullizomic is given by 2n-2, trisomic is given by 2n+1, and monoploids, they are single set of chromosomes denoted by X. Question number 38. The method developed by Heyman to estimate the various components of genetic variance using 6 population is, So it is called generation mean analysis. It involves several generations from crossing between two inbred lines, which provides the estimate of epistatic effect under chromosomes. The ability of an inbred to transmit desired performance to its hybrid progenies is, so the correct answer is option number C, combining ability. In case of general combining ability, it is the breeding value of parents and is linked to additive gene effects. In case of specific combining ability, it is linked to non-additive gene effects, which include dominance or epistatic effects. The structure of chromosomes can be seen at, so it is clearly visible in the metaphase. Question number 41. Heterosis can be easily fixed in. So it can be easily fixed in apomictic crops. It is normally broken due to segregation, but apomixis produces the plants as a clone of mother plant, and it overcomes the problem of breaking the heterosis. Question number 42. The ratio of economic yield to biological yield is described as, so it is described as the harvest index. Question number 43. The heterosis estimated over the superior parent is called, it is called heterobeltiosis. The term was given by Blitzer and the formula is by F1 minus the beta parent upon the beta parent whole multiplied by 100. It gives the amount of the heterobeltiosis. Question number 44. Norrin 10 dwarfing gene is responsible for dwarfness in, so it is responsible for the dwarfness in case of wheat. Also, the RHT1 and RHT2 gene, they are also derived from the Norrin 10, which improve the lodging resistance. Question number 45. Single gene affecting more than one character is termed as, so it is termed as pleiotropy. Question number 46. The condition of missing a segment of a chromosome is called, it is called deletion. Here we can see duplication is when a segment duplicates itself. Deletion, as we have discussed, the missing segment. Inversion, here the segments interchange the positions. In case of translocation, one segment is in another place and the other segment comes to the other place. So the translocated the places. Question number 47. The synthesis of messenger RNA is called, so it is called transcription. Question number 48. Number of chromosomes in the endosperm is, so it is 3n. Endosperm is triploid, which develops by the fusion of polar nuclei, that is 2n, with the sperm nucleus, that is n, through the double fertilization process. Question number 49. The pollen that has no fertilizing effect on the stigma of the same flower is called, so the process is called the self-sterility. In case of hercogamy, we can see the position of anthers and stigmas, they are different. In case of dichogamy, the stigmas and anthers, they mature at different times. So that's why the self-pollination is not achieved. Question number 50. The smallest functional unit of a gene is called, it is called a recon. Cistron is the section of DNA or RNA that codes for a specific polypeptide. Mutin, it is the smallest unit of chromosome which occurs in mutation. Question number 51. Match the following. Pure lines, they have a narrow genetic base. Multilines, they have a broad genetic base. Isogenic lines, they have one gene difference. Pleiotropy, it has manifold effects of a gene. Then backcross, it has a donor parent. Question number 52. Match the following Oryza species with the set of genomes. So Oryza sativa, it is diploid having AA genome. Oryza grandiglumus, it is a polyploid variety having CCDD genome. Oryza australiensis, it has the genome EE. Then Oryza officinalis, it has genome CC. Oryza brachyantha, it has the genome FF. Question number 53. Match the following. T.S. Venkatraman is related to nobilization of sugarcane. K. Ramia, a rice breeder. B.P. Pal, the first DG of ICAR. Pushkarnath, we know, a potato breeder. Satyatham is a cotton breeder. So that was it. Thank you.